good day, everyone, and welcome to the second OBI webinar, today on the topic of spines and motif participation. My name is Michael Reimann from the Open Brain Institute, and I am joined today by Oliver and Theodora from Frontiers Journals. Maybe say a quick hello to show your faces. Have they joined yet?
Hi everyone, it's nice to meet you. I'm Oliver, I'm here with Theodora, and we'll just provide you with a little bit of information around our Frontiers journals at the end, I think.
Great, thank you. Theodora, do you also want to say a few words?
Hello everyone, it's very nice to see you today, and as Oliver said, I look forward to showing you some information from our journals.
Yes, today I will show you how to investigate the question, are dendritic spines that participate in non-random connectivity motifs larger than others? But more generally, this is really about techniques to study the structure and topology of connectivity and spines in context with each other. And so this is the agenda for today. I will start by giving an introduction on the topic in terms of the scientific background, followed by a live demo on how to study the topic. And then, as mentioned, we will hear from the Frontiers journals. And should I succeed in getting you interested and you choose to follow this kind of research, they may be an excellent choice to publish your results. And at the end, I will answer all of the questions you might have. And the way this works is that during the presentation, when you have a question, you can enter it in text form, and this will be sent to our moderators. And then at the end, we take a note of it and we'll answer all of those questions. So some disclaimers on what to really expect from this webinar and what I want you to take away. So the high-level idea is that we can relate the structure of local networks to the anatomy of spines to better understand how the networks form. And the specific angle I choose are synaptic connections that form large non-random motifs placed on larger spines than connections that do not. And that is only one possible angle, an interesting one as we will see, but mainly I want you to understand that this type of analysis really has become possible and maybe even quite easy with modern data sets and tools. And it must be said that for the sake of time, I will have to glance over some of the mathematical details of the analysis. There is some mathematics involved. However, to anyone that is really interested in the details, I can give full access to all the analysis, the source code, and the resources that underlie my presentation. You can get access on the OBI page. Then you can look at the code and understand all of the details and all of the explanations. If you have registered for the webinar, and you probably have because you're here, then after this is over, you will receive an invitation email that gives you access. And so this is a high-level overview of what I will be showing. So we will find the connectivity matrix of an electron microscopy connectome on the OBI pages, and we will use it to identify a number of neurons of interest based on their participation in connectivity motifs. Then we will skeletonize the neurons, which means we will extract a machine-readable representation of their morphologies, including all their spines as individual elements that we can analyze. The morphology is placed in the context of the circuitry, meaning that we know exactly which synaptic connection is innervating which spine. And that allows us to investigate two questions. First, do neurons that participate in a specific position of a connectivity motif differ in their spine anatomy from neurons that participate in another position? For example, is their spine density lower? And second, do individual connections that form part of a large non-random connectivity motif, do they form larger spines than connections that are not part of specific motifs that are more randomly formed? And given all of this, I will now introduce some background information about connectivity motifs, spines, and how they may be related. So let's begin with motifs. So data on local connectivity is often expressed in terms of connection probability. The probability that a randomly picked pair of neurons is synaptically connected. And this kind of implies that connections in the circuitry form randomly and independently. A motif is a group of neurons that are connected to each other in a specific way, such as this example here, which is a cycle where one neuron is connected to the next, which is connected to the next, which is connected to the first. And if connections form randomly and independently with a given probability, then we can easily calculate how many instances of each motif we would expect to be present between a given number of neurons. However, it has been consistently and repeatedly observed that in actual biological circuitry, some of the possible motifs are massively more common than expected. And this indicates that there are other non-random mechanisms in the formation of connectivity. And there are many potential mechanisms and results and theories about this. But on a high level, we can distinguish between two types of mechanisms. There's targeting, where the growth of individual neurites like axons is guided to specific cells or classes of cells. And selectivity, where the neuron shape or morphology gives rise to a set of potential connections, and then more complex non-random mechanisms select specific cells that are active, specific connections that are active. And of course, these are not mutually exclusive, and the truth can be a mixture of both. So in terms of targeting, what can we say? Well, it's a clear observation that the effects of targeting, since they are mediated via the shape of neurons, would be visible not only in the connectivity, but also in the shape of the dendritic and axonal trees. And yeah, indeed, if we look at different classes of neurons, they tend to have very different morphologies. But this difference may also be unrelated to connectivity and have a different reason or purpose. Or maybe it relates to connectivity only on a more coarse-grained level. So we can ask, we can ask, if we predict connectivity from morphology, does motif overexpression emerge? And this has actually been tested. You can predict connectivity from morphology by placing a large number of reconstructed neuron morphologies at adequate locations in a virtual volume. And then you place connections where the axons meet the dendrites, or at least at a subset of these axodendritic touches, where the subset is selected without considering motifs or the larger connectome structure. Then what the authors did is they counted the instances of a specific class of motifs called directed simplices. Now, also later on in the main part of this demonstration, that's the type of motif we will be considering. So try to pay attention, please. A directed simplex is an all-to-all connected motif with directed feed-forward connectivity. We see some examples here. Formally, we can define them recursively, where a zero simplex is simply a neuron. A one simplex is a connected pair. And then we can define even larger types of simplices with more neurons, as I said, recursively. Specifically, an M simplex, where M stands for an integer, is a combination of a K simplex and an M minus K simplex, where each neuron in one part has a connection to each neuron in the other. So a one simplex is a synaptically connected pair. If we take two of them, like in this example here, 0 and 1 form a one simplex, 2 and 3 form another one simplex, and then we have connections from each neuron here on the left side to each neuron here on the right side, forming a 3 simplex. Now, the reason why this class of motif is interesting is that we can have simplices of different sizes. And the larger the motif is, the less likely it is to emerge purely by chance. So neurons and connections in larger simplex motifs may on average be more strongly affected by the mechanisms that shape non-random connectivity. And therefore, by contrasting simplices of increasing size, we can take a peek at the mechanisms and maybe learn a little bit about them. Now, back to non-random connectivity from morphology, where the authors counted the counts of these motifs of different sizes, and they found that a prediction from morphology here on the right and various biologically measured connectomes provide very, very similar results, which indicates that neuron shape, without any further selectivity mechanism, at least explains some forms of motif expression. But of course, there are also mechanisms of selectivity and rewiring, which brings us to spines. Spines are certainly a very important element of neural circuits, as the majority of excitatory cortical connections are formed onto dendritic spines. By increasing the neuron surface, they may increase the number of connections a neuron forms. But another view is that they increase the number of potential connections that a neuron can choose from, and thereby enabling it to be selective about the partners it connects to. And we know for sure that spines are related to the formation of connections, where a dendrite, here shown in green, you can actually observe how it grows outwards towards an axonal bouton, shown in red, and contacts it and forms a synaptic connection. But there is indication that this is not everything, and that this meeting of spine and axon really first only leads to a silent synapse, which is then further filtered. To quote from this publication down here, multiple filopodia, that is essentially spines, from the nearby dendrite locally sample the adjacent neuropil for potential axonal partners, and successful connections are then selected for survival based on coactivity with nearby task-related spines, ensuring that the new spine preserves functional clustering. Now, this describes selectivity based on coactivity, but the connections selected that way may also be the ones that form the non-random motifs. The same filtering criteria are then the ones that further strengthen the synapse through long-term plasticity, growing the spine even larger. And if all of that is the case, then there should be a correlation, logically, between the spine anatomy, or more precisely its size, and participation of connections in motifs. That's a hypothesis, which until recently would have been very difficult to study because this idea of motifs and motif participation requires a very zoomed out view of circuitry. You need multiple neurons and their connectivity. And spines are a very zoomed in view of individual elements of connectivity. However, it has been made possible through enormous efforts through electron microscopy connectomes, such as the Microns data, which provides information on connectivity between thousands of cells. At the same time, as highly detailed morphologies and shapes of individual neurons in the circuitry. And this includes, as you can see, the spines. The only issue is how, this view makes really no distinction between the individual spines and the rest of the neuron, making it really difficult to analyze individual spines and their shape. But in a few minutes, I will show you how you can automatically separate and extract all spines of a neuron as individual shapes. Which brings us to the demonstration part of the webinar. So as a reminder, we start here. We will find the Microns EM connectome on the OBI pages and analyze it to find neurons of interest. So we move to the OBI virtual labs. So as I mentioned, you will get an invite link. And if you follow that link, you will be joining the spines and motif participation project on the Open Brain Institute virtual labs. And this is what you will be seeing. So roughly speaking, on a high level, these virtual labs are split between data, workflow, and notebook tabs, where data refers to a database of various forms of neuroscience data, such as electrical recordings, neuron shapes, or models and simulations. And workflows are standardized neuroscience workflows that use the data as input, such as a workflow for defining and launching a simulation or others. Notebooks lead to a deployment of Jupyter notebooks, which are completely integrated with the rest of the page and give you seamless access to all of the data under the data tab, as we will see in a bit. And this is a good starting point. We have placed here some exemplary notebooks to get started with the various types of data. But also, this is where we can place project-specific analysis, such as 3 notebooks that I've placed here, which are specific to this effort to understand spines and motif participation. So we begin by finding the connectome that we want to consider, which is of type circuit, because it defines neuronal circuitry. So it's under the data model tab, where we have various data points of circuitry. And you will notice that this comprises both circuitry that has been built by statistical modeling algorithms, but also experimentally measured circuits, where we find it is very important to represent these 2 different types of data in the same format to really enable researchers to compare them. But that's for a different webinar. Right now, we're interested in the Microns data, which we see here. So this is a representation of the connectome of the Microns data set. Now, what we do with it, I have implemented in a notebook, as mentioned. So we begin with the notebook select motif participating neurons, which we simply run. Yes, so we've run it. There's a lot of theory here, which I will be skipping over, as I mentioned. But as I said, you can have access to all of this, and then you can read all of this at your own pace. So logically, we start with importing certain packages that we need. But then the next part is authentication with the OBI platform, which allows us to gain access to the various types of data that I've shown you. And we select the project context in which we want to work, which is this SpineSet Motif Participation project, which gives us access to the data of that project. So if you work on these pages, your data is compartmentalized and only visible to members of your team until you choose to make everything public, which hopefully you would do when you publish it. Then we will next download the adjacency matrix of this Microns connectome. And yeah, for that, we need to now find and access the data point that I've shown you before. And there are various ways of doing it. There is a search functionality in code. So we have really code packages that you can use to search for data based on its properties. But since we already know what we want to analyze, which is the Microns connectome, we can simply copy its identifier string and just fill it in here, which is, in this case, easier, which then allows us to access the data, as you see, and load the connectivity matrix, which we've done. So the next step is now we have to restrict the analysis to a subset of the neurons which have been manually proofread. Well, we don't have to, but I think it's the proper thing to do for maximum accuracy of the results. And additionally, I filter here to only consider excitatory neurons because it's just inhibitory connectivity is formed according to different principles. So we exclude it for now. And now we find, and we can now find neurons of interest based on their motif participation. So a little bit more on that. So what we will be doing is we will, as I mentioned, consider directed simplex motifs. Here's a reminder what they look like. They are densely connected in a feed-forward way. And because of this feed-forward directionality, the neurons participating in the motif are actually very different in terms of their connectivity. For example, the neuron in position 0 is a sender of connections to all other neurons, and the neuron in position 3 is a recipient of connectivity. So what I'm trying to say is we can sort them from left to right, such that there's always a connection from a neuron on the left to a neuron on the right. And now my idea is that this may manifest itself in their spinal anatomy, that neurons in this position may be different than neurons in this position. Now what we do is the following. A neuron can participate in multiple simplex motifs. In this example here on the right, we have two simplices. Like one is formed by these 3 neurons, one is formed by these 3 neurons, and one is formed by these 3 neurons. And already by this, you can see that in all 3 simplices, always the red neuron is participating in all of them. So in position 0, we really only have this one neuron that is participating heavily, in contrast to this position over here, where 3 different neurons are participating. And so this is what we will next do in the notebook. We count how often each neuron participates in each position of a 5th simplex, so much larger than what we're seeing here. And then we find the top participators, and those are the ones we analyze further. Now, this might seem complex, like combinatorially, you have to find all combinations of this particular motif, but really it runs very quickly. We have very optimized algorithms for that. And so in this connectome, we have 112 5 simplices. And here we see the identifiers, the local identifiers of the neurons participating in the various positions. And you can see that one and the same neuron can participate in several of them. And from there, it's very simple to just count how often each neuron is participating in each position. And yeah, we see like there is this exemplary neuron here, a layer 5 ET neuron, which is participating in 33 5 simplices in position 0. That is one of our neurons of interest. And now just this data frame, we just save to a file for keepsake. So as a reminder, what we have done are the first 2 steps. We have found the data that we want to analyze, and in a notebook, we selected motif participating neurons. Now we want to look at those neurons and extract their morphology and spines. That is on the page under workflows. That's a standardized workflow. Under process data, we have EM cell mesh skeletonization, where we now select simply the neuron that we want to have the morphology of. So we have here the identifiers of those neurons, and we can just pick one, just copy its identifier, making it easier to find. There, that's the one which we use for a skeletonization task. We need to just give a name to this task. And there are some additional parameters one can play around with, but the defaults are pretty solid values. We can then generate the skeletonization tasks, and then we can launch them. Now, this task would take around 15 to 20 minutes to complete, which of course, we don't have time to sit here for 20 minutes and wait for the results, so I won't do this now. I have already done it. So now we pretend I've done this 16 times for the 16 neurons of interest, and we can find the results afterwards under data within our project as a morphology. Here under morphology, we now have the morphologies of the 16 neurons of interest that we have identified. You can look at them. We get some basic statistics of them and a basic 3-dimensional view. You see it's a beautiful pyramidal cell, no slicing artifact. It's a complete morphology. But we were promised spines, and I can show you spines. For that, we have for visualization of the spines a separate notebook where we can run this. And once again, I've already prepared this to speed things up. The notebook to visualize skeletonized morphologies, including the spines, because I've promised you spines and you will see spines. It just takes a few seconds to launch. There it is. Now all spines at once, it's just too heavy, so we can only look at the spines section by section, but we can select a nice section with a lot of spines. And we can see here that we indeed have the spines isolated as individual elements. You see they're in different colors indicated, really showing us that individual spines have been extracted and are now accessible to us for analysis. And a different section gives us the spines of that section. Okay, so but now the next step. Now what we want to do is not just stare at spines, but do a proper analysis. Once again, that is implemented within a project notebook, which I called Analyze Spine Count and Size Against Motif Participation. Once again, it can be launched by using the run functionality. And once again, I've already prepared this to speed things up because running this notebook would take around 15 to 20 minutes. It's already open here. where again, we start by first loading the data we created in the previous analysis step. So the data on the 16 neurons of interest and how much they're participating in each position. We authenticate with the platform, select the webinar project to be able to access our 16 morphologies with spines. And in this case, actually, we do not need to copy the identifier for those morphologies one by one into the notebook. We use here the search functionality to find the spiny morphologies based on their identifier automatically. And as you can see, if we do that, we find the 16 skeletonized morphologies with spines, which we can then load. And then having loaded them, we can plot their basic morphology without spines. This is such a beautiful example with an axon going over to a nearby column. But we also have access to the spines, most importantly, programmatic access to the spines. So here I'm simply picking one of these 16 neurons and a randomly selected spine of it. And as you can see, we have information about where the spine is, and we have its shape, its surface mesh extracted, which can then be subjected to analysis. So as a quick reminder where we are, we have skeletonized and visualized the output of this step over here, where we have run the workflow EM mesh skeletonization, allowing us to look at the output of the workflow, which is really morphologies with individual spines that we can analyze. Now we will go to our first actual scientific question, which is once again in a different notebook. And yeah, this is the question, do the neurons at different positions of the motif have different spinal anatomy? And so we continue here. We do not have only access to the individual spine meshes, but we also have a spine table, which is simply a big table with an overview of all the spines on this morphology. So this neuron has 4,500 spines, and we can get an overview of basic anatomical parameters such as orientation in space, etc., etc. But we begin with a very simple analysis, spine density, which simply requires the spine count for the 16 neurons and their dendrite length, and then you just divide one value by the other, giving you the overall spine density, which we have run here. So now we have added to our table of the 16 neurons dendrite length, spine count, and spine density. Then the last thing before we can start plotting data is I calculate what I call the significant simplex position for the neurons, which is simply, as you can see here, these columns give you the participation of the neurons in different positions of the simplex motif. And these are 5 numbers. I just want to condense them down to a single number. And so I simply calculate the average position for a neuron. So this example, which participates in position 1 and 2 and 3, has an average position of 1.76. It's really just to shrink this down to a single number, because then we can simply plot this simplex position against the spine density and get a negative result. I mean, at least that's what it looks like to me. It looks like the overall spine density, while highly variable, is not different between neurons in their different simplex positions. Just for your information, the colors in this plot indicate the layer of the neuron that is considered, where the message is simply that this motif comprises neurons not of a specific layer, but of all possible layers in all possible positions. So that was a negative result, but we can also plot the total spine count against simplex position, and we find a more positive result with a positive correlation, and even this outlier, you know, the one with a high spine count being having an average simplex position of 5. So we can ask, is that significant? Where we find, based on this p-value, that I would say, yeah, this is a significant correlation between this simplex position, the position within the motif, and spine count. But we can do even more. Since we have access to individual spines, we can calculate the surface area of each and every spine of each and every neuron. That is the reason why I chose to run and execute this notebook beforehand, because this is something that iterates over hundreds of thousands of spines, yeah, tens of thousands, and takes a while. But it's very simple. With a few lines of code, we can calculate the size, the surface area of all of those spines, and we can see that it really looks like neurons participating in the in this particular motif differ in their surface area based on their position, where the more you are towards the receiving end of the motif, that is receiving inputs, the larger the spines get. And that is also a significant result. So going back to our overview, we have run this notebook where we have found a result that the position within the motif does not really get you differences in spine density, but in spine size. And now we will go to the second question, which is moving from analyzing differences between individual neurons and then calculating the grand average statistics of their spines. You saw we did a mean over thousands of spines. Now we're looking at the individual connections that are part of the motif and ask, is it specifically those connections that are on larger spines? The next question. That's the next question. And in order to ask and answer that question, we need more information. Right now, we have already a lot of information. We know for the neurons their spines, but we also need to know the information about their synaptic connectivity in the context of the spines. And that is a slightly different data type under the data tab within the OBI webinars. We call this type of data a synaptome because it comprises not only the morphology of the neuron and its spines, but also all incoming synapses of the neuron. So we know for this, not only the for this neuron, not only the spines, but also which connection is innervating which spine, allowing us to do even more. In the notebook, I call spines and edge participation. Again, we go to that notebook, spines and edge participation. So enjoy my beautiful drawing. So what we're doing here is now we're considering these simplex motifs of different sizes, as I mentioned. So the whole cartoon here is a 5 simplex. But if we take just 3 neurons of it, it would be a 2 simplex. If we take 4 neurons of it, it would be a 3 simplex, and so on. And what I mentioned is that participation of a connection in such a 5 simplex motif is indication really of something non-random ongoing, because in statistically generated connectomes, you really never observe that. Whereas participation in a 1D simplex has little meaning. It's simply a synaptically connected pair of neurons. And so we can ask for each connection, what is the largest simplex motif that it's a part of? How high up does it go? For example, in this cartoon, the edge here in blue is really only part of a 1 simplex. whereas the edge in yellow is part of a two simplex formed by this neuron, and this neuron. And by the way, about all of this, I in the very first notebook, I have some, if you need more information about these motifs, in the very first notebook, I put some valuable references. So edge participation. Again, we run the notebook starting with some imports. We load the previous data. We authenticate with the platform. We download the microns data and analyze it. And this time we calculate a slightly different, we use a slightly different function called edge participation, which also runs very quickly. And this is what it gives us. This gives us a nice table with one row for each synaptic connection within this proof red part of the connectome, which tells us for each connection the source and target neuron, and the simplex motifs it's participating in and how many. So we see that this edge really is only part of a one simplex and nothing larger. No indication for anything larger. Whereas this one over here is additionally part of 9 two simplices and 6 three simplices and so on. Then we load one of these synaptome entities that I showed you a while ago, a little bit ago. We start with analyzing one of them, and then later on we do this for all 16 of them, for all 16 neurons, where we load that synaptome of one exemplary neuron, which is has this node participation. So this neuron is participating in position 5 of 9 of these motifs. And we load the connectivity of that neuron, as I said, all incoming connections, giving us this beautiful table where it tells us that this neuron has an in degree of over 9,000 incoming synapses. And for each of them, we know which spine it is on, indicated by the spine ID. Is it on a spine or a shaft at all? And what is its afferent, its presynaptic partner? Additionally, after one more step, we also see the surface area of the spine, or if it's on a shaft, the value is 0. And I have combined this with the edge participation I calculated earlier. So now we see for those connections also the simplex motifs they're part of. Having one example here, we're on a fairly big spine, which is actually part of even a 5 simplex motif. And this allows us to simply plot the size. So what is the largest simplex motif that the connection is part of against the surface area? And we see a positive overall correlation. So meaning if a connection really is part of one of these large motifs, the larger the motif is, the larger the spine is. And we can also plot the probability that it is formed on a spine at all. And this is the result that absolutely surprised me, which is that the larger the motif, the less likely the connection is on a spine. I'm surprised by that. I would have expected, if anything, an inverse. But maybe this is just that one neuron. So we repeat that analysis for all neurons, which takes a few seconds. That's why I've already prepared this. I've already run this. And now we can plot the same result as before for all neurons. So each line now is the result for one of the 16 neurons that we have identified before. And we again plot the size of the largest motif they're part of against the surface area. And we find overall, I would say, subject to more rigorous statistics in the future, I would say overall we find an increasing trend. However, the variability between neurons with respect to this measure is larger than the increase we see from simplex size 1 to 5. So it's an effect, but it seems rather weak. And there are also some neurons where this is going down. It's, I think, 3 neurons where this is going down rather than up. The second result, spine probability. Again, we do for all neurons. And here we find something that is maybe more convincing. It really seems to be the case that for all 16 neurons, the larger the motif, the simplex motif, I must say, the simplex motif that the connection is part of, the lower the probability that it is on the spine. So now we have also tackled the second question, which I think gives a nice overview of, in general, what is possible with these tools with respect to correlating the structure of connectivity to the anatomy of spines. A brief summary: synaptic connections that form non-random motifs differ in the ultrastructure of their postsynaptic target from connections that do not, where neurons that receive inputs in a motif, so they are in the receiving position, tend to have more spines than neurons that send outputs, and neurons that receive inputs in non-random motifs also tend to have larger spines than neurons that send outputs. And the second notebook result, connections that are part of a non-random feed-forward motif tend to be formed onto larger spines, but again with an asterisk. And more solidly, connections that are part of non-random feed-forward motifs are more likely to be formed on a shaft. Now I really think here you can do this analysis as well with all of the tools we make available. Some things that you can think about is interpretation of this result. I'm still puzzled. What do these results mean? Also, what I've looked at were neurons that were specifically selected to be top participators. But what does the average neuron look like? You can, of course, look at a different type of motif. This was a very narrow look at one specific class of motifs. You can also look at inhibitory neurons and, how do they work with respect to spines? I only looked at excitatory. You can consider the different classes of neurons in the context, pyramidal cells, as I said, versus inhibitory neurons, etc. But what I think is exciting is this enables some really advanced hypothesis testing. So just think about it. There are these important theories of microcircuit function, the roles of individual layers and how they are interconnected and how that implements function. And in principle, these theories should be able to give you predictions of which connections are more central to the theory, are more important. And then you can hypothesize that they should be potentiated and have larger spines. You can test this. So these are just some ideas. And with that concludes the live demo part of the presentation. So I hand now over to Oliver and Theodora to tell us about where you will publish your results on this topic.
Thank you, Michael, and thank you for the demonstration. It was very interesting to see. But hi, everyone. It's nice to see you all, and I hope that you're well. I'm Oliver, and I'm here with my colleague Theodora, and we're going to speak a little bit more about Frontiers and Frontiers journals. So I'm the journal specialist for Frontiers in Computational Neuroscience, and for those less familiar, the journal focuses on advancing our understanding of the brain through computational neurosciences. This includes everything from biologically realistic simulations to AI-driven methods for understanding mental health conditions. The journal is led by chief editors Si Wu and Misha Sodykes, and alongside a global editorial board of experts across computational and theoretical neuroscience. In terms of what we can offer you as a journal, there are several ways to get involved and that you can contribute. So first of all, of course, we welcome article submissions, and we have several different article types to offer. So if you have an article that's ready to be submitted, feel free to reach out to us, and we're happy to support you in finding a suitable location for this. Secondly, we also offer the opportunity to host your own research topics. which are curated collections of articles around a given theme. We find that this is a really good way to shape discussion in your field, build collaboration, and it's a way for you to also put your research interests at the forefront of discussion. And then finally, we're always looking at ways to expand our community. And there are also opportunities to join our editorial board. So if you want to contribute more directly to the peer review process and the journal's direction, we'd be very happy to discuss an editorial board position with you. Finally, we're also very proud to partner with the Organization for Computational Neurosciences, and we're actively working with the research community through initiatives like society-led research topics here. So if you'd like to host a research topic in collaboration with the Organization for Computational Neurosciences, we have the QR code on the screen. Feel free to access this QR code. It should take you to a form where you can register your interests, and we'd be very happy to reach out to you about that. But yeah, thank you very much for listening. Feel free to let us know if you'd like to get involved, and I'll pass over to my colleague Theodora. Thank you, everybody.
Yes, thank you, Oliver. And hello, everyone. My name is Theodora, and I'm a journal specialist with Frontiers in Neuroinformatics. Our journal is an open access journal as well, which was launched in 2007, and it is led by Jan Bialy from the University of Oslo, Michael Denker from the Jülich Research Center, and Sean Hill from Sun Science in Switzerland. The journal currently has an impact factor of 2.5 and a site score of 5.5. And if you have your phone nearby and would like to explore the journal directly, you can scan the QR code on the right-hand side of the screen, and it will take you straight to its homepage. In terms of scope, the journal focuses on neuroinformatics research that helps the community analyze and share neuroscience data. The main subjects of interest include applications and technologies that facilitate data sharing, the development of data and knowledge bases across all levels of neuroscience, work involving existing clinical or basic neuroscience databases, novel informatics approaches that merge clinical and basic neuroscience data, and new tools for data acquisition, analysis, visualization, and dissemination of nervous system data. We accept many article types as well, which you will see listed here. So from original research to systematic reviews, methods, hypotheses and theory, technology and code papers, mini reviews, data reports, but also quite recently, we also accept Fair Square articles, which combine a data management and publishing service all in one workflow. At this time, we have 8 article collections open to the journal, covering topics from EEG and artificial intelligence through FAIR neuroinformatics more broadly, to ethics and privacy in AI-driven neuroinformatics, or even neuroinformatics for youth mental health. And finally, we are actively looking for new collaborations, and we welcome discussions around joining as associate editor, community reviewer, or topic editor, as well as discussions on how we can support you in submitting a manuscript or perhaps a Fair Square data article. As highlighted earlier, we also accept Fair Square data articles, which are the first article type of their kind, designed to give datasets the same recognition as traditional research papers. So each one is peer-reviewed, citable, and AI-ready, and comes with an interactive data portal that lets others explore, analyze, and reuse the data all in one place. So if you would be interested in any of these types of collaborations, please get in touch through the email address on the slide, or feel free to reach out to me directly at theodora.constantinescu@frontiersin.org. And with that, I thank you very much as well for your attention.
Thank you. Thank you, Theodora. Thank you, Oliver. Yeah, this gives us some more minutes for Q&A. Were there any questions?
Is there a way to identify the cross-sectional area along a spine?
Yes. As I said, you have access to the spine surface mesh, and for example, you could a mesh is really a representation of points, and then triangles are defined as triplets of the points. But so essentially what you can do is use those points. You can load them as a NumPy array, identifying the main axis of the spine. There are algorithms to do that. And then you can calculate the distance of the points from this main axis and calculate the area of, the area that they essentially delineate. That is, I mean, it's algorithmically, there are a few steps involved, but it would be fairly simple to do. And the algorithms to do this have already been developed. I know that some of our collaborators from Ilan Segev Lab have a paper on this topic, so you can find that in the literature. And in the future, we may even, I've shown you as part of this the spine table, which comprises a list of all the spines and some basic morphological information. And we might even, if people tell us that this is useful, we might even include things such as the neck diameter or the head diameter or equivalent in the spine table. And then you don't need to worry about this. And it's just a number that you can directly access. And the second question?
And the second question, where can we find the Python notebooks?
Yeah, the Python notebooks. So, I'm still sharing. So again, so the way this works is over here. Again, you presumably you will receive the invitation email. You follow the link, you log in, yada, yada, yada. After that, you're landing here. Then the notebooks are over here. And so these are this notebook section is split into two parts, public and project. So public is where we, OBI, put some broad examples about how to work with the data types. These are more educational. So where you want to go is project, which are the three notebooks that that I've shown you. Yeah, well, there they are. You can press for each notebook on the plus button. And you can either access you can see what this is about. You can download it if for some reason you want to run it locally. Or you can just click run, and then again, this opens Jupyter Hub and opens the notebook. And that's the ways in which you can find the notebooks. Were there any more questions? Maybe in the meantime? Well, I guess maybe questions come once people join the project and try to try things out. Again, if you join the project, you try things out in each of these notebooks. As you can see here, I put my email address, and yeah, don't hesitate if you don't get it. If you have questions, to just write me an email.
Maybe you could say something about simulating these models, Michael, and linking them to function.
Oh yeah, I mean, that's another thing that you can find under workflows. We can build single cell models. So where this is another potential use of these skeletonized morphologies, where we can actually, again, this is the list of the project morphologies. These are the 16 neurons of interest. We can actually build something simulatable out of them. So I have to move this button around. So again, this is one of these beautiful neurons. And yeah, this is actually maybe a very interesting thing, because a lot of single cell modeling is done with morphologies from slices, which always have cut off dendrites. Here we can build a single cell neuron from them by selecting the cell and selecting an electrical model that describes the firing behavior of a neuron. So for example, let's find a good model for a pyramidal cell. Let's take this one. And after a brief compatibility check, we should be able to build and simulate. Nope, I've checked. I used the wrong E model. This is a bit impromptu. That's why I haven't prepared this. There we go. So so I can now just build a model which now we can actually simulate. So this is another interesting avenue of things you can do where, I mean, we have given these neurons, we have speculated that they have different roles based on their position within these motifs. Now we can actually simulate them and see if there are any specific differences in that regard. So we could now set up a stimulation protocol, determine what voltages we want to record, and so on and so on. So maybe there are some interesting differences between these neurons, also in their electrical behavior. All right.
The later webinars, yeah.
Yeah, again, we have more webinars in the future. Oh, there's a question now. Where can I find the question?
Why the first email?
Yeah, why the first email? Of course, you make one mistake and people immediately pounce on it. Why? So somebody asked, why did the first e-model not work? Presumably because it was a model for an interneuron, and this is a pyramidal cell with, it has apical dendrites, it has basal dendrites, and an interneuron does not make that distinction. And within the within the electrical model, there is a clear distinction. And so if you want to use an E-model that that does not make that distinction together with a morphology where the distinction is relevant, then they just technically cannot work. More questions? Okay.
Sorry if you covered this, my internet dropped at the end. Is
Oh.
there a way to identify the cross-sectional area along the spine?
Yes. Okay. The cross-sectional area along the spine. And the answer is yes. Yes, absolutely. It's, as I mentioned in, let's find the notebook. Let's find the tab. In this notebook, as I mentioned, you have access to the spine mesh. And if I just drop a new cell here, spine mesh dot vertices. So these are literally points on the surface of the spine, and you can use these points to identify first, the main axis along the spine, and then the diameter at each point along this main axis algorithmically. There are algorithms to do that. Currently, we don't have them prepared and implemented in the notebook, but it would be fairly simple to do.
Could you share where do we find the I?
The IPyNB, so the IPyNB file of what we have on the screen now, this, okay, that's very simple. Again, it's under notebooks. And then it's a project-specific notebook. And this was analyze spine count and size versus motif participation, which downloading it downloads as you can see, a zipped file, and the zip file contains the IPyNB file and all the requirements like images that are embedded, etc.
Back to the previous, where should we check whether it's an interneuron or pyramidal?
Yes, so that goes back to the very first notebook, which I should still have open somewhere. Select motif participating neurons. So this is actually an interesting question. So already in the Microns data, because it's an amazing data set managed by amazing people, they have already done quite a lot of classification of those neurons, and especially of the neurons that are part of this proofread data set. And so that is also programmatically accessible. And when you run this notebook, as you can see, there's the list of the 16 neurons, and each of them have been given a M type according to this classification. So it's layer 3A, so that's a pyramidal cell in layer 3 with a thick tuft. This is L5ET, that's an, I think, extratelencephalic projecting neuron, and so on. So for the details on the classification, read the various papers by the Allen Institute on the Microns data set. But as you can see, none of them are interneurons. And why are none of them interneurons? Because one of the first things I did with the data was I said, please, I only want to analyze excitatory neurons. More questions? Well, at least we've had a few. In that case, I would say we can end it here. So everybody, thank you for your attention. Thank you for your questions. And yeah, except if you want the invitation, the access to those notebooks, and to all of the data that I have demonstrated. So have a good rest of your day, everyone. And I see you hopefully in a month in the next webinar. Bye.